Welcome to Methodical Medics. In the previous videos, we've covered glycolysis and the transition reaction. Now, in this video, we'll be covering the citric acid cycle step by step. And towards the end, I'll show you some memory tricks to remember each intermediate and enzyme. So if you are in a biochemistry class or taking the MCAT, this would be perfect for you. All right, so here's an overview of the citric acid cycle. It's also called the tricarboxylic acid cycle or Krebs cycle, but citric acid cycle is the most common name. So there are 8 intermediate compounds and 8 enzymes. Its purpose is not to produce ATP directly, but instead to load electron carriers like NAD and FAD. And when they're loaded, they will move on to the next step, which is oxidative phosphorylation. I will be making a detailed video on that shortly. So the citric acid cycle is not just a step in carbohydrate metabolism. It's also a part of fat and protein breakdown and kind of acts like a central hub that connects all the domains of metabolism. Anyways, let's begin with step 1. So we start off with the 2-carbon acetyl-CoA from the bridge reaction. Citrate synthase is an enzyme which condenses acetyl-CoA with oxaloacetate, a 4-carbon molecule. These together form a 6-carbon citrate, which is what the cycle is named after. Right up here, you can see where the carbon-carbon bond is formed. And if you'd like to know the exact reaction mechanism, let me know in the comments and I can make a follow-up video showing you it. This is one of 3 irreversible steps in the Krebs cycle. And each of these steps are often a point of regulation. In this case, citrate synthase regulates how much acetyl-CoA enters the cycle. Step 2 is a reversible isomerization reaction. This means that the chemical formula stays the same, but the chemical structure changes. In this case, citrate is turned into isocitrate by a conatase, moving an OH group from the center carbon to an adjacent carbon. And also from now on, if you see a single bonded red oxygen atom, assume it's in an OH group. This is to avoid visual clutter of adding in all the hydrogens. Step 3 is a major part of the Krebs cycle, and it's definitely worth memorizing. Isocitrate dehydrogenase catalyzes the first decarboxylation and oxidation step. First, isocitrate is oxidized. NAD is reduced to NADH and a proton, and when it does this, the same OH group that we moved in the last step becomes a carbonyl group. So it loses a hydrogen and gains a double bond. Next, it decarboxylates and forms carbon dioxide, which is a waste product. So now we have a 5-carbon alpha-ketoglutarate. This is a key regulatory step of the cycle. Isocitrate dehydrogenase activates when energy is low, so when there's like a high presence of ADP, or is inhibited when energy is high, when there's high ATP or high NADH. Step 4 is another and the last decarboxylation step in the citric acid cycle. Alpha-ketoglutarate loses a carbon dioxide, forming a 4-carbon intermediate. And then coenzyme A, the same one that was used to make acetyl-CoA, is attached to it, forming a 4-carbon succinyl-CoA molecule. NAD is reduced again to form NADH, and interestingly, the alpha-ketoglutarate dehydrogenase enzyme has the exact same reaction mechanism as the PDC in the bridge reaction. So check out my video on that. I go in great details explaining that. And this is also the last irreversible step in the TCA cycle. It gets a lot simpler from here. Step 5 is very simple. Succinyl-CoA synthetase converts succinyl-CoA to succinate. I highlight a sulfur bond in yellow, which is very high in energy. When this breaks and coenzyme A dissociates, the energy is used to drive the phosphorylation of GDP into GTP. And GTP can be used directly as energy, or it can convert into ATP depending on the needs of the cell. In step 6, succinate is converted into fumarate by succinate dehydrogenase. Fumarate has the exact same structure, but it has a double bond between C2 and C3. Also, electrons are transferred to a different carrier this time, FAD. FAD is reduced into FADH2, and it's different from NADH because it doesn't carry as much energy, which will be shown in the next video. Next is a hydration reaction catalyzed by fumarase, and it adds a water molecule, so a hydroxyl group, to the fumarate, turning it into malate. It's a very simple step. Finally, malate is oxidized, turning the hydroxyl group to a carbonyl group by malate dehydrogenase. This generates another NADH and a proton, and notice how we regenerated oxaloacetate. Well, congratulations. We've walked through the citric acid cycle and we're back at step 1. I know it's quite a lot to memorize, but there are memory tricks to memorize the enzymes and intermediates that I'll get into shortly. This is a quick and classic mnemonic, which has helped so many students over the years remember the intermediates of the Krebs cycle, including myself when I was writing my MCAT and biochemistry tests in general. Can I keep selling sweets for money, officer? Each letter represents an intermediate. Citrate, isocitrate, ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and then oxaloacetate. Once we remember the 8 intermediates, then we can memorize the 8 enzymes. We can do this by building a lopsided ladder and then fill in each enzyme. So if you've noticed, a lot of these enzymes have their name attached with the intermediate it acts on. So there are 2 unique enzyme names that don't follow this rule. Aconitase, which turns citrate into isocitrate, and fumarase, which works on fumarate to turn into malate. Next, we have the 2 S enzymes, which is citrate synthase and succinyl-CoA synthetase. Citrate synthase doesn't directly act on citrate. It acts on oxaloacetate and acetyl-CoA to form citrate. But it's okay to build the ladder this way because it still follows the correct order. Just be aware that it doesn't really follow the same naming scheme like the other enzymes. And now finally we can fill in the last 4 parts of the ladder, which are D enzymes or dehydrogenases. These all follow the naming schemes correctly, so there's nothing to worry about there. And if it's a dehydrogenase, it is also an oxidizing step. So just remember that. All right, to summarize what we've been through, we start off with 1 glucose molecule, which turns into 2 acetyl-CoAs. These 2 acetyl-CoAs will undergo 2 revolutions of the citric acid cycle. And it will produce a total of 6 NADH and 2 FADH2 molecules, which will go on to be harnessed in the electron transport chain. We will also get 2 GTPs, which will be used as energy or converted into ATP. And we'll also have 4 carbon dioxide molecules, which will be exhaled as waste. Thank you so much for watching my third video. I will see you in the electron transport chain. Let me know in the comments if you'd like to learn anything else.